Hello everyone, my name is Dr. Kaushal Shah and today I'm here to discuss the answer paper of Cyclic Test 2 biology subject for 9th standard ICSE board. Right, so let's start with this. The paper was for 30 marks. Let's start with the first question. The first question was fill in the blanks. Let's discuss something about the fill in the blanks here. So fill in the blanks, one word answer, straightforward answer, certain rules that you're supposed to follow while writing the answer of fill in the blanks in your answer paper. You're supposed to write the complete fill in the blank with the question itself and underline the correct answer, right? Okay, so let's start with the first one. Centrosome is found in dash. The answer is cytoplasm. It is, a part of cytoplasm where it is present. So that is what is a clear answer. And if you write that properly, underlining cytoplasm, 1 mark will be given to you. Right? Let's go to the next question, people. That is dash provides turgidity to plant cell. The vacuoles are the one which provides turgidity. So the answer here is vacuole. And again, you're supposed to write the entire sentence underlining vacuole. The third one, cell membrane is made up of dash and dash. The answer is lipids and proteins. Right? Commonly, we call cell membrane as a lipoprotein membrane because they are made up of two things. Lipo stands for lipids, proteins. So that is our lipids and proteins. Mind you, people, this entire fill in the blank, which has two blanks, is for 2 marks. That is how if you see the first question fill in the blank, which is 4 blanks are asked, but the marks are 5 because here there are 2 blanks, right? So this one is of 2 marks. Next question is dash is the structural unit of life. The correct answer is cell. So that is what you're supposed to write in the blank. So very simple questions about fill in the blank. One single word answers are supposed to be written, right? Along with the entire statement and underline that answer. Now let's go to the second question, and that is name the following, which is again for 5 marks. Here there are 5 questions. So here you are supposed to write, it's 1 mark each. You're supposed to write functions, a name of each of the following questions. Let's start with the first one. The first question is about a cell without nucleus, right? And the answer to that is prokaryotic cell. Now, in name the following, if you don't write the question, that's fine, right? You're supposed to write the answer properly. So when I talk about prokaryotic cell, we know it's a primitive type of cell without a well-defined nuclear membrane, without a distinct nucleus, right? So we call it prokaryotic cell. The next question. The next question is about the largest cell. When I talk about the largest cell, the answer that you can write is ostrich egg or bird's egg, and in bracket you can write ostrich egg, but do not just write ostrich, right? That is very important because the cell in ostrich is very small. The largest cell is egg of an ostrich. So you can write ostrich egg, egg of an ostrich. That is the correct answer. Next question is about a structure which help in the release of energy inside the cell, right? So yeah, it is mitochondria. We know mitochondria is a powerhouse of the cell because it is the one which releases energy. So that is what should be your answer, mitochondria. Next one, the fourth one is bearer of heredity. Now when I ask you about bearer of heredity, name the following. The correct answer has to be chromosome, right? If you write genes or DNA, it is not exactly correct because genes are what are known as hereditary units, to be very specific, right? So when bearers of heredity comes, the correct answer has to be chromosomes. The last one in the name the following is the site for protein synthesis, right? The correct answer are ribosomes. Ribosomes are the one which manufactures proteins, and that is why they are known as the sites for protein synthesis. Okay, let's go to the third question. Let me just take it down so that we can understand it better. Just a minute. Yeah. So now we're going to the third question, which is state the role of the following. And these are 4 questions for 1 mark each, right? So let's try to understand something about this role of following. When I say role of following or functions of following, they are a similar kind of questions, right? So let's try to understand something about this. The first one was nucleus, right, in the question. So let's try to understand. Now it is 1 mark each. You can just write one function, that is the role of that particular organelle, and that would be done. right? So if it is a nucleus, you can write it is present, suspended in the cytoplasm. It is located in the cytoplasm. It suspends itself there. And when it comes for the role, it controls all the activity of the cell. So we say all the activities of the cell are controlled by the nucleus. Now, in your answer, if you just write that, because the question is just role of the following, if you just mention that yes, nucleus controls all the activity of the cell, it is still correct. You will get 1 mark, right? Okay, let's go to the next question, and that is about cell wall. Now, when it comes to cell wall, we know that cell wall is present only in plant cell, right? It is absent in animal cell, and it occurs as a protective covering surrounding the plasma membrane, and its main function is protection, right? So if this question is about role, here there are some additional points given for the answer, but if it is just cell wall function or role that is asked, you can easily write that it is protection to the cell, right? The next one, the third one, is mitochondria. As we just discussed in the previous main question, that when I talk about mitochondria, it is the one which provides energy in the form of ATP. And whenever you write ATP in bracket, always prefer to write the full form also. That is adenosine triphosphate, right? And this energy, that is ATP, are used for various metabolic activities or functions inside the cell. If you want to write some additional information, you can mention that yes, the mitochondria is also known as a powerhouse of the cell, right? So that is all about, I would say, mitochondria. Now I'm going to the next question, and that is about ribosomes. So it's a kind of answer which is already there. If you know the earlier question answer, you can easily answer this. So ribosomes function, they are the sites for protein synthesis. If you write this much only, still you will get full 1 mark, right? And as they produce protein there, we also call them factory of the cell. So that was question number 3. Now let's go to the next question, and that is question number 4, which has distinguish between. Now in distinguish between, you're supposed, see, it's a 6-mark question. There are 3 distinguish between which are asked. So ideally, 1 distinguish between is for 2 marks. And for that, you're supposed to write 2 points each. When I say 2 points each, if this is a question here about plant cell and animal cell, you're supposed to write at least 2 points in plant cell side and 2 points in animal cell side, right? Here we have mentioned many more points so that you have an option of writing, but you can write any of the 2 points, right? And you'll get full 2 marks. Now, some rules that you're supposed to follow for writing the answer of distinguish between. Then we'll start with the question, because this rule will be applicable for all the distinguish between answers, right? So let's try to understand. Whenever distinguish between question is asked, you're always supposed to make 2 columns, give the heading properly, and then start writing your answers, and that too always by giving numbers to the points, right? Because if it is a 2 mark, you're supposed to write 2 distinguish between. If it is for 3 marks, you're supposed to write 3 distinguish between, right? So that is a basic rule that you're supposed to follow. Now let's start and answer the first one, and that is distinguish between plant cell and animal cell. A very important question, very easy one also. So let's start understanding something about the points of each of them. Starting with plant cell, usually they are larger with distinct outlines. We know, right? As they have a protective layer, what is known as cell wall, there's a distinct outline which is there. In animal cells, usually they're smaller in size as compared to plant cells. They're oval and with less distinct boundaries. The reason being absence of cell wall, right? The next point, cell wall is made up of cellulose, and that is present in plant cell. In animal cell, we know cell wall is absent. Okay. Third point, vacuoles in plant cell, they are very large, and there are 1 or 2 in number, but huge vacuoles as compared to, if we compare that with animal cell, vacuoles, if any they are present, they are very few and very small in size, right? So that is again 1 more point. Let's go to the 4th point, and that is cytoplasm is less dense in plant cell, right? And as compared to that, if we go to animal cell, here it is not only just dense, but it is granular. When I say granular, in animal cell, the cytoplasm contains a lot of granules, right? The next point is something about, again, cytoplasm. Cytoplasm is in the form of thin lining pushed to the periphery, right? Guys, understand one thing. As we know that vacuole are the largest organelle in plant cell, they occupy a lot of position, and that is why everything else is pushed towards the periphery of the cell, including the cytoplasm. And animal cell, cytoplasm fills almost the entire cell. Why? Because it is no, there are no big or large vacuoles which are going to obstruct the cytoplasm. Next point, Golgi bodies are diffuse in the plant cell and are called dictyosomes. Right? Now we know specifically they are known as dictyosomes only in plant cells, and they are diffuse in the plant cell. As compared to that, if we go to animal cell, Golgi bodies are well developed and present in front of the nucleus. They are very close to the nucleus normally, and that is how they function in animal cell. Next point is something about centrosome. When I talk about centrosome, it is absent in plant cell, but it is present in animal cell. And now comes the last point, and that is plastids are present in plant cell. We know plastids are present only in plant cell, right? And in animal cell, they are totally absent. So these are 8 points of distinguish between plant cell and animal cell. When it's a question for 2 marks, you're just supposed to write 2 each, right? Okay. With that, now let's go to the next distinguish between, and that is something about, I would say, centrosome and chromosome. Here we have just mentioned 2 points because there are hardly any point of differences here, and that is why it's for 2 marks. Let's try to understand something about them. So centrosome, it is clear area of cytoplasm which is generally very close to the nucleus, and this is the organelle from which spindle fibers develop during cell division, right? So that is what is the centrosome all about. As compared to that, if we go to chromosome, chromosomes carry hereditary information or genes, right, which transmit genetic characters from parents to offspring. So it is this chromosomes which are nothing but the hereditary materials, you can say, in the form of genes. They have some kind of materials, and that is what carries all the characters from parents to offspring. As you can see, centrosome, chromosome, totally different overall, right? Now let's talk about the second point. The second point is centrosome is found only in animal cell, right? They are not present in plant cell at all. And if we compare that with chromosomes, chromosomes are found in the nucleus of both animal cell, plant cell, right? So yeah, that is 2 simple distinguish between points for centrosome and chromosome. So that, now let's go to the third question there. And the third question is about distinguish between cell wall and cell membrane. Again, very simple question. And here we have mentioned 3 points. You are supposed to write 2 if it is for 2 marks and 3 if it is for 3 marks. So let's start with cell wall. Cell wall is a non-living rigid layer, right? It's a non-living rigid layer which is present only in plant cell. That will come later. You can also mention it here itself that it is present only in plant cell. And when you talk about cell membrane, it is a living, thin, and a flexible membrane, right? There's a kind of flexibility in there, and that is how there might be some kind of movement of cytoplasm also, which can be easily done. Okay. Next point, cell wall is made up of cellulose. Cellulose is a complex type of carbohydrates, people, and that is what makes the cell wall very tough and rigid. Okay. Now, when I talk about cell membrane, it is made up of lipoproteins, or what is also known as lipids and proteins. Lipids, in simple words, are known as fats, right? The third point, cell wall is freely permeable. When I say freely permeable, it allows anything and everything to pass through it easily without any kind of hindrance or obstruction, right? But when I talk about cell membrane, it is semi-permeable, or what we also called it as it is selectively permeable. When I say selectively permeable, it will not allow everything to pass through the membrane. It will only allow selective things to pass through the membrane, right? So that is something about the distinguish between cell wall and cell membrane. Let's go to the next question. In fact, the last question there, and that is about answer in brief and any 2. It's for 10 marks, right? Let's discuss something about that. So first question here is, what are prokaryotic cells and eukaryotic cells? Give one example each. So here, first of all, we are supposed to explain what are prokaryotic cells and eukaryotic cells along with one example, right? So let's start with prokaryotic cells. So as it is clearly mentioned here, prokaryotic cells do not have a well-organized nucleus, right? When I say they do not have a well-organized nucleus, why do we say that? Because the nuclear material in the cell is not enclosed by a nuclear membrane, a kind of covering which is needed, right? So in prokaryotic cell, if there is no nuclear membrane, if there is no boundary to the nucleus, you cannot say that it has nucleus, right? It's like a room with no walls is not a room, right? There has to be walls to consider it one particular room. Here also, without nuclear membrane, the nucleus is considered absent, right? Okay, the nuclear region without a nuclear membrane is called nucleoid, as we don't call it nucleus, but it is like nucleus, so we call it nucleoid. Okay, the chromatin material is represented by just one single circular DNA molecule. So when I talk about that genetic material, chromatin material, it is just one chromosome, one single circular DNA molecule is present, right? Further, let's go to other features. The cells here, I'm talking about prokaryotic cells, lack membrane-bound organelles like endoplasmic reticulum, right? Golgi complex. Mitochondria are still present, but membrane-bound are not present, and lysosomes, nucleolus, etc. So all the advanced organelles which are found in, you know, advanced cells, they are completely absent in prokaryotic cell. Bacteria and blue-green algae are the examples of prokaryotic cells. So that is all the description about. That is about all the description of prokaryotic cell with its example. Now let's go to eukaryotic cell. When I talk about eukaryotic cell, it is an advanced cell, modern cell, right? So it is a well-organized cell with a proper well-organized nucleus. When I say well-organized nucleus, here the nuclear material is enclosed in a proper nuclear membrane, right? And these cells have well-developed membrane-bound organelles. So all the organelles are present along with a proper covering to each and every organelle, which is why we call it membrane-bound organelles. And all the different organelles are like endoplasmic reticulum, lysosomes, chloroplast, mitochondria, etc. And when I talk about examples of eukaryotic cell, examples, all plants and animals, all higher plants and animals have eukaryotic cells. Here they have mentioned about fungi, protozoa, all these have what are known as eukaryotic cell, right? Okay, let's go to the next question now, and that is about draw a neat labeled diagram to show the structure of plant cell. So yeah, this is what was taught, that when it comes to diagram, you'll have to see on the screen a proper plant cell. And one thing that you'll have to do always when it comes to biology diagrams is practice by yourself at home, right? So please practice your diagrams. I'll just tell you some important points that you're supposed to focus when you draw a diagram for your exam. One, the diagram has to be neat, right? If you draw plant cell, it should appear like plant cell and not animal cell. That is how it is. Okay. First thing, it has to be neat. Second thing, it has to have proper labelings, right? There has to be proper labelings. Then the labeling part and the diagram part should be connected with lines, right? Preferably just lines. That is what you're supposed to do. Next thing, each and every diagram should have a title, right? What diagram it is. If it is a plant cell, you are supposed to mention that it is a plant cell. See, that is missing here, but you are supposed to mention it. And then lastly, each and every diagram should be put up in a box, right? So these are certain rules that you're supposed to follow while drawing the diagram, any diagram for biology, right? So as you can see, this is plant cell. We have mentioned about cell wall, then comes plasma membrane, or what is known as cell membrane, cytoplasm, right? Then you talk about vacuole, nucleus, mitochondria, chloroplast. These are some important organelles which you are supposed to show in the plant cell, right? So that is what is shown here very clearly. Okay, now let's go to the next question, and that is the last question that we are supposed to discuss, and that is something about the figure which is given in the question paper, and you are supposed to label all the essential parts and organelles according to their numbers which are shown in the figure, right? So let's try to understand each one of them one by one. The first one is what is cell membrane, or you can also write it as plasma membrane. It is the outer covering of this cell, and as you can see, this cell doesn't have a cell wall or something like that. It doesn't have a fixed rigid shape, and that is why we can say it is an animal cell. And when we talk about animal cell, the outermost covering is nothing but cell membrane or plasma membrane. Okay, now let's go to the next number, and that is number 2. Let's go in order so that it becomes easier for you to understand. So if you see here, I'll just try to move the cursor here so that you can understand this. I'm talking about this particular number 2. right? If you see this particular numbering, it is showing endoplasmic reticulum, but there you can see that dot like structures. That means it is rough endoplasmic reticulum. So you are supposed to very clearly mention about rough endoplasmic reticulum. Now, why do I say that you are supposed to mention it very clearly? Because the next one, number 3, let's see that, and it is here. I'll just show you. See this. I'm just moving the cursor here, and if you see here, you will be able to appreciate that here basically they have what is. Let me just show it to you with a highlighter if it comes. Okay, yeah. So see, this is what I was talking about. Number 3, right? If you observe this carefully, they have shown again endoplasmic reticulum here, but this time they do not have that dot like structure. And I hope you guys remember that dots are nothing but ribosomes. So your ribosomes are not present, and that is why it is smooth endoplasmic reticulum, right? Okay, let's go to number 4 now. This structure here, see number 4, this kind of labeling, and you see it. This is something that there are two things, and they are very close to the nucleus. They are almost perpendicular to each other, and they are nothing but centrioles, right? So I've told the features earlier, and then I've named so that you guys can understand it properly. Let's go to number 5. Number 5 is one very easy one, because the structure of mitochondria here is very unique. It is present in the cytoplasm, and that is where the number 5 labeling is mitochondria. Now we are talking about number 6. Let me just show you here. See this, number 6 here. When I talk about number 6, this is something inside the nucleus. And if you're observing it carefully, it is pointing to that thread-like structure, the scattered thread-like structure inside the nucleus. And that is nothing but chromatin fibers, right? Okay, let's go to number 7, which is on this side, left-hand side. And it is showing some dots in the cytoplasm. When it is showing some dots in the cytoplasm, we are referring to granules, right? Granules, small spaces where storage of starch or other kind of food or something is done. So that is what is granule. Now let's go to number 8, which is on the right lower side here. I'll just show it once again. And that structure, if you observe it carefully, right, is Golgi body. You can write Golgi complex, Golgi body, or Golgi apparatus. Everything is correct. Just to let you know, Golgi apparatus appears like, the Golgi body appears like deflated balloons. There are series of deflated balloons which, are placed one after another. That is what is a kind of appearance of Golgi bodies. Then let's go to number 9, and this 9 is the one which shows the entire covering of the nucleus. So this is, they are pointing out to the nucleus here. So the very simple thing, it is generally in the center, spherical organelle. That is what is nucleus. And then comes the last one, and that is, number 10. Let me show that. This one, people, and that is the cytoplasm, right? So that is all about the labeling part of this particular question. And with that, we are done with the discussion of answer paper of Cyclic Test 2 for 9th ICSE board. And that's it. Thank you so much for listening. All the best.